Hello and welcome back to the Biochemistry for Health Sciences channel. We will continue our discussion on the applications of enzyme kinetics. And today we'll try to answer the question, do enzymes change in diseased tissues? In previous videos on enzyme kinetics, we discussed how to obtain three very important values, Vmax, kcat, as well as Km. So do enzymes change in diseased tissues? Let's look at a publication from 2015. This paper came out in Cancer Cell International, and this publication title is Kinetic Characterization of Lactate Dehydrogenase in Normal and Malignant Human Breast Tissue. As many of you might know, breast cancer is the most common cancer in the world. Approximately 13% of new annual cancer cases is breast cancer. Breast tissue is very rich in adipocytes, these cells that synthesize and store fat. Of course, there is connective tissue elements such as collagen, and the rest of the breast has ducts which end as lobules, and this is where milk is synthesized and then transported out to the nipple area. The most common type of breast cancer involves these cells here, these epithelial cells that line the ducts. So let's look at this epithelial cell and discuss the difference between a normal ductal epithelial cell and one that is cancerous. Like all cells in the body, the epithelial cells, these ductal epithelial cells, require energy at all times, so they need to synthesize energy all the time. The most important source of fuel for energy production for most cells is glucose. The best way to get maximum amount of energy, so energy is in the form of this chemical called ATPs. So to get maximum amount of ATPs or energy per glucose, that is about 30 to 32 ATPs per glucose, the glucose will have to go through different metabolic pathways. So let's trace these pathways that glucose have to go through in order to get maximum amounts of energy. So first, the glucose will have to go through this metabolic pathway called glycolysis. At the end of glycolysis, we get pyruvate, and then this pyruvate will enter the mitochondria of the epithelial cell, of the ductal epithelial cell, and there the pyruvate will be converted into acetyl-CoA by this enzyme system here called pyruvate dehydrogenase complex, or PDH. The acetyl-CoA will then enter the Krebs cycle, and out of the Krebs cycle we'll get hydrogens, which will be carried by these molecules, NADH and FADH2, and these hydrogens will be transported into the electron transport chain, where they'll react with oxygen to form water, and out comes a lot of energy, and this energy is then used by the enzyme ATP synthase to synthesize ATP from ADP and PI. So let's just call this system here the oxidative phosphorylation system, or OXPHOS. So going through this route will give the epithelial cell about 30 to 32 ATPs per glucose. So when oxygen is sufficient, if there's enough oxygen available to the cell, since you can see here oxygen is required for this oxphos part here. So when oxygen is available to the cell, then the glucose will go through this whole system that we just discussed, and that will give the cell 30 to 32 ATPs per glucose molecule. However, when oxygen is insufficient, if the epithelial cell doesn't have enough oxygen, then only this pathway will be used, glycolysis. Okay, because this pathway will give you 2 ATPs per glucose. So we get only 2 ATPs, but this is a much shorter pathway, glycolysis, and it doesn't require any oxygen. So this is what this ductal epithelial cell will do. If oxygen is available, then it will go through the whole pathway of glycolysis, PDH, Krebs cycle, and the electron transport chain oxidative phosphorylation to give you 30 to 32 ATPs per glucose. And if oxygen is not available, then the cell will use glycolysis, and that will give you 2 ATPs per glucose. So with this background, now let's look at the enzyme that is called lactate dehydrogenase, or we'll abbreviate it as LDH. So what does LDH do? Now LDH can convert this pyruvate into lactate. So pyruvate reacts with NADH to give you lactate and NAD, or LDH could interconvert in the other direction. It could convert the lactate into pyruvate by reacting with NAD. So lactate plus NAD could give you pyruvate plus NADH. So this enzyme will catalyze or can catalyze the reaction either going in this direction or in this direction. So when the cell does not have enough oxygen, when it does not have enough oxygen, so now the cell is going to rely on glycolysis to give it 2 ATPs per glucose. Okay, so now the cell is only going to rely on glycolysis if there is no oxygen available. So this part here would not happen. Now, if the cell was only going to rely on glycolysis, then it would have to convert this pyruvate into lactate. And why does it have to do that? Because by converting pyruvate into lactate, the NADH is converted into NAD. And why do we have to do this? Well, this NAD can be used right here in this step number 6 of glycolysis. So glycolysis requires a lot of NAD. Now NAD is derived from a vitamin, niacin. So we do not make this vitamin, we get it from our diet. So since it comes from a chemical that we do not make, to make sure that we have sufficient amounts of NAD, we basically recycle. So this NADH that is produced in step number 6 is converted by lactate dehydrogenase into NAD, and this NAD goes back and it can be used by reaction 6. So in this way, LDH helps sustain glycolysis. So the aldehydes reaction sustains glycolysis, allows glycolysis to happen to make sure there is always enough NAD available so that glycolysis can continue and so that the cell can get its 2 ATPs per glucose. So that is what a normal ductal epithelial cell in the breast is going to do. Okay, so just to summarize, if there is oxygen, it's going to use the OX4 system to generate energy that will give you 30 to 32 ATPs per glucose. Without oxygen, it's going to use glycolysis to give you the 2 ATPs per glucose. And to do the glycolysis part, it has to make sure that the pyruvate is converted to lactate so that the NADH is converted to NAD, and the NAD can be used by glycolysis in step number 6. Now, in breast cancer, when the epithelial cell becomes cancerous, a very strange thing happens. When they study these cancer cells, the cancer cells prefer to use glycolysis so that they can get their 2 ATPs per glucose, although the cancer cell may have enough oxygen. Okay, so although there's enough oxygen, the cancer cells will still prefer to use glycolysis to generate their energy. Now, so this is kind of quite different from what we saw in a normal cell. So this phenomenon is called aerobic glycolysis. So glycolysis is happening in these cells although oxygen is plenty. Although there's a lot of oxygen available, these cells are using glycolysis to generate their energy, not the OX4 system. Okay, so this was discovered by the scientist Warburg, and this effect is also called the Warburg effect. So this strange phenomenon that we see in cancer cells, what we call aerobic glycolysis or the Warburg effect, this effect has been known for many, many years, and is associated not just with breast cancer, but many, many different types of cancer. So in this publication, the authors looked at this enzyme, lactate dehydrogenase, LDH. They looked at this enzyme in normal breast tissue as well as in malignant breast tissue. So here's some of the important data that is summarized in this table. So on the right-hand side, we have the cancer cells, and on the left-hand side, we have the normal cells. So when you look at this reaction of LDH going from pyruvate to lactate in this direction, we see that the cancer cells have a lot of active enzyme, 4,000 compared to 1,747 when pyruvate was used as the substrate for lactate dehydrogenase. When NADH was used as the substrate for lactate dehydrogenase, we are talking about 2,788 compared to 1,370. So cancer cells have a lot of active enzyme that converts pyruvate to lactate compared to normal cells in the breast tissue. The Km for the lactate dehydrogenase was not much different. When pyruvate was used as the substrate for normal tissue, the Km was 0.63. We use the word S0.5 because this enzyme shows sigmoidal kinetics. So instead of Km, we use S0.5. So the S0.5 was 0.63 for normal. For cancerous cells, it was still quite close, around 0.78. When NADH was used as a substrate, the S0.5 is 0.33 for normal tissue, and for the cancer version of the tissue, it was 0.30. So not much difference here, but a lot of difference in terms of how much active enzyme the cancer cells had compared to normal cells. So when the reverse reaction was studied, so here lactate plus NAD, the reverse reaction will give you pyruvate plus NADH. So when the reverse reaction was studied, this is the data we get using lactate and NAD. So as far as the specific activity, how much active enzyme there was, no difference. No difference between normal tissue and cancerous tissue. As far as the Km was concerned, the cancer cells had higher Km for lactate as well as for NAD. So much higher Km for the cancer cells compared to the normal cells. So all of this data basically showed that the breast cancer tissue had LDH that was better in converting pyruvate into lactate. So it was converting pyruvate into lactic acid, okay, rather than converting lactic acid into pyruvate. Because remember, this enzyme LDH can catalyze the reaction in either direction. So cancer cells prefer to make pyruvate into lactic acid and not the other way around. So this makes sense when we look at the Warburg effect. The fact that cancer cells prefer to get their energy from glycolysis. They prefer to get their energy from glycolysis. That means cancer cells prefer glycolysis. Although they might have a lot of oxygen, they still prefer to get their ATPs from glycolysis. And to sustain glycolysis, the pyruvate must be converted to lactic acid. Okay, so by converting pyruvate to lactic acid, the cell is also regenerating NAD, converting NADH to NAD, and this NAD can be reused in step number 6 here of glycolysis. So all this makes sense, and that's why cancer cells have LDH that prefer to convert pyruvate into lactic acid. Now, as far as the question, why do cancer cells prefer aerobic glycolysis? Why do they prefer to make their energy through the glycolysis system rather than the OX4 system? Although the OX4 system gives you much more ATPs per glucose, yet these cancer cells prefer to use the less efficient glycolysis system. Now, why they do this, we do not exactly know, but there are several suggestions that we find in medical literature. So one possible reason is that the glycolysis system of generating energy is much faster. It's much faster than sending pyruvate into the mitochondria and then going through PDH and then going through Krebs cycle and then going through the electron transport chain and then the ATP synthase. So compared to going through the whole system, this glycolysis, just going through glycolysis, is much more rapid. Okay, much more rapid. So although the yield is less, in a given time, because this is much more faster, in a given time it's possible that the cancer cells may be generating as much energy as going through the whole system simply because it's much faster. Another suggestion is that since cancer cells are multiplying very quickly, they are making new cells, proliferation very quickly, they need a lot of carbon building blocks. They need these raw materials that are necessary to make, for example, nucleotides and proteins and lipids that are required for the new cells. Okay, so they need to make a lot of new biomolecules for the new cells so that they can sustain their cell division, the cell proliferation. So in this respect, glycolysis is better because in glycolysis, glucose is converted into this carbon raw material pyruvate, and this pyruvate carbon material can be converted into many other raw materials that can then be used to make all kinds of biomolecules, including nucleotides, amino acids, and lipids. However, if the cell used this system, the Oxford system, the whole glycolysis, PDH, Krebs cycle, and the electron transport chain, if the whole system was used, then the carbon, in this case, the carbon comes out as not as a building block, it basically comes out as a gas, carbon dioxide. Okay, so it's harder to make things from carbon dioxide rather than to make things from pyruvate. So this is the second suggestion that is available in medical literature, that cancer cells prefer the glycolysis, aerobic glycolysis, because it can get more carbon building blocks for synthesis of biomolecules that are necessary for the new cells that are made. Now, a third suggestion by scientists is the acidic microenvironment. Now, glycolysis, as we mentioned, produces pyruvate, but this pyruvate has to be converted into lactate so that the NADH becomes NAD, and this NAD can be used in reaction number 6 to sustain glycolysis. Now, lactate is really lactic acid, This is an acid. So by generating lactic acid, the cancer cells are basically producing acid. And remember, acid can denature proteins nearby. So it's possible the cancer cell might be secreting acid into the surrounding so that it can loosen itself from other cells, and it can also loosen the connective tissue so that it can separate from the other cells and can start spreading. Okay, metastasis. Okay, so one of the features of malignancy, of malignant cancer, is that these cells can separate from other cells, the cancer cells, and then loosen the surrounding connective tissue and start migrating. They start migrating towards some sort of blood vessel or some sort of lymphatic vessel so that they can spread. So the generation of lactic acid basically gives the cancer acidic microenvironment, and this acidic microenvironment will enhance or will stimulate metastasis, the spread of cancer. Another suggestion is that using glycolysis and not using oxygen is going to change the reactive oxygen species, the ROS, the free radicals. So the amount of free radicals, so this free radical balance is changed. And by changing this free radical balance, it's going to alter cell signaling. So basically, by not using oxygen and by producing acid, the cell signaling is going to change into a format that is going to promote the growth of the tumor. So this is a very active area of research. Whether one or more of these effects is happening in cancer is very important for us to know, because if we understand what the cancer cells are doing, then we could be able to better design drugs that could stop or reduce the cancer metastasis as well as the tumor growth. So the answer to the question, do enzymes change in diseased tissues? Yes, in breast cancer, it does change for example, in this example, the kinetics of the lactate dehydrogenase enzyme, and this may also happen for many other diseases. Okay, we shall discuss another application of enzyme kinetics in the next video. Until then, have a wonderful day.